Hello everybody, my name is Dr. Evan Gordon. I'm a neuroscientist working at the Waco VA. My specialty is using neuroimaging techniques, and particularly magnetic resonance imaging, that's MRI, to study how traumatic brain injuries, TBIs, affect the brains of veterans. So what does this look like in practice when we're using neuroimaging? So we take a veteran patient who's suffered a TBI. We put them in our MRI scanner, slide them on in, and we image their brain. And we can get a very nice, very detailed picture of the brain of this individual who has suffered a traumatic brain injury, and we can try to figure out what the TBI has done to their brain structure. We can also use functional MRI techniques, FMRI, which is a very exciting technique that lets us look not only at the structure of their brains, but also at the activity of different parts of their brains. And we can use this to try to get a really good picture in this person of what brain regions are active, how the TBI may have affected which brain regions are active. Once we get a very nice picture of brain activity in this person, we can do a technique called group averaging, where we get a whole bunch of people. Maybe they've all suffered a traumatic brain injury. We look at their activity levels. We smush them all together, average them, say, okay, in general, across TBI, people who've suffered a TBI, where do they have brain activity, where do they not have brain activity? We can compare these maps to, say, similar maps we get from healthy controls, and we can say, okay, the TBI tends to affect these brain regions. I've been doing this sort of research for 10 years, and this is the way we do it. This is the way the field does it. We average the brains together, we compare the groups to each other. And this group averaging approach has been really useful. It's been incredibly informative to the field of neuroscience to help us understand how brains in general work. We've learned about where in the brain, what parts of the brain are needed to perform working memory, holding information in your mind. We've learned about what parts of the brain help you move different parts of your body around. And we've learned about what we call the networked organization of the brain. And I have a nice picture here where we know that the yellow parts, the parts of the brain I've colored here yellow, tend to talk to the yellow parts, and the parts that are red tend to talk to red parts. And this is very exciting and helps us understand how the brain is organized. At the same time, this group averaging approach, I've been thinking about this a lot over the last few years. This group averaging approach can be so limited. And the reason is that this brain organization varies across people more than neuroscientists want to admit. So let's say we show people a picture of a face. We know that there's been tons of research. There's this particular area at the bottom of your brain called, we're incredibly good with coming up with names, it's called the face area. And it becomes active when you see a face. Here you can see the places in the brain that become active in different people. So somebody has this spot here that's in green, somebody has this spot here in purple, somebody has this spot here in red. What you can see is that this is the same general area of the brain. We're looking at the bottom of the brain here, but they don't really overlap that well. It's not the exact same spot. There's variability from person to person in exactly where their brain responds to face. And this is a problem, because this means that this group averaging data, as fantastic as it is for helping us understand, okay, the bottom part of your brain responds to faces, it's problematic when we want to say something about a particular individual patient. So if we do this neuroimaging experiment, let's say we are doing it in somebody who has post-traumatic stress disorder, and we're showing them fearful faces. And we see this one person who has PTSD, and they seem to have a weak activity in this general area to this stimulus. What does that mean? Maybe their brain didn't activate. Their brain is not activating enough to fearful faces, and that must be the cause of, that must be related to their PTSD. But there's another explanation that this image really points at, and that's that maybe their brain activity is just in a different place from the place where most people's brain activity is. And this is really problematic, because what we found is that this group averaging of neuroimaging data, this approach, it's not clinically relevant. And this has been really bothering me over the last few years. I think fMRI is the best technique we have for understanding the function of the living human brain. I've been doing fMRI work for 15 years now. I'm still amazed. I can put you in a scanner. I can watch how different parts of your brain are becoming active. It's amazing. It's like magic. This is such a powerful technique. It should be able to provide us with clinically useful information. I want information about diagnosis. I want information about prognosis. I want to be able to generate treatment recommendations for individual patients who have traumatic brain injury, who have PTSD, who have depression, who have schizophrenia. And for 25 years, since we developed fMRI, it's mostly failed at doing that. We've learned a ton about how the brain works, how it's organized. We have not been able to help people clinically. How much of that failure might be due to the fact that we haven't been focusing on individuals? We've been doing this group averaging approach. Do we even know how to use fMRI to study individuals? And I think we don't. So we have really been pushing in the last few years, together with our collaborators in St. Louis, to generate an individualized neuroimaging paradigm, really completely different from the way that neuroimaging has been used previously. The goals are to develop techniques to use fMRI for individual-focused analysis. Let's be able to say something real and concrete about an individual patient. This approach, it's very different. Instead of having 1,000 people and we look at where the brain activity is on average, we have just a few individuals, but we collect very large amounts of data in each individual. That lets us maximize the precision of our measurements. And what we want to do is we want to, once we figure out how exactly we can do this individualized neuroimaging approach, we want to take the techniques that we've developed, the lessons we've learned, to the study of veterans with traumatic brain injury and post-traumatic stress disorder. So we have started to do this work in healthy controls. We did a project with our academic collaborators where we scanned 10 healthy controls. We scanned them for 2 hours, and then we scanned them for 2 hours again, and we scanned them for 2 hours again. We did this 12 separate times. Each person was scanned for 24 hours. We collected 4 hours of structural MRI, 5 hours of what's called resting state MRI, where you just lie there and do nothing, 5.5 hours of MRI while they're doing various different tasks. This is a lot of time just lying in the scanner. We've started to see some really interesting, really amazing things. I pointed out to you before that we know about the network organization of the brain. The yellow areas tend to talk to the yellow areas. The red areas tend to talk to the red areas. When we take all our 10 subjects and we average all their data together, we get a picture that looks pretty much exactly like the picture I showed you before. This is the network organization of the group average brain. But when we look at individuals, they look a little different. We still see the big yellow areas and the big red areas, but we're also starting to see a lot of features of the individual's brain that are specific to that person. I'm just pointing out a few here. We see this little piece of this purple brain network here where there's nothing in the group. This little piece of this green network here and here where there's nothing like that in the group. This red network has this little stripe that runs down here. There's nothing like that in the group. This is because this person is different from other people. When we average all these people together, the specificity that we have in this person gets washed away because it's being averaged across a bunch of people who each have specific little pieces, but are not common. They're not exactly the same. They get averaged out. A really important question that we have to ask is how much scanning is needed to get these reliable measures of brain function in each individual. If we want to make a picture like this, if we want to be able to say what's special about this person, what's happening in this person, how long do we need to scan people? Do we need to scan people for 24 hours? What we did is we took our data and we did what's called a split half analysis. We cut the data in half. We said, okay, we have a picture of their brain in the first half, we have a picture of their brain in the second half. How similar are those two pictures depending on how much time we're using? And it turns out each one of these lines is a separate person. It turns out in each person, once we start collecting enough data, we get very, very high reliability between the first half and the second half. Once you get about 60 minutes, an hour of scanning, you start to have reliability up around 0.9, which is very, very close to perfect reliability. This is really pointing at the fact that we need maybe an hour of MRI scanning to get a good picture of somebody's brain activity. How much scanning have have people been getting? Around here. That's not so good. And that may be why we have not made a lot of progress in getting clinically relevant measures of brain function. So takeaways from this individualized neuroimaging approach, we know that the brain organization now does vary across individuals, and that when we average these pictures of brain organization and function across individuals, we lose features that might be important. And we can collect accurate individual-specific measures of brain function if we get enough data. So the next thing we wanted to do was we wanted to use this high-data, individualized neuroimaging approach to understand traumatic brain injury and post-traumatic stress disorder. So, of course, we know that TBIs are common among military veterans, and we know that having a TBI can result in persistent behavioral and cognitive issues. We also know that having had a TBI increases your vulnerability to PTSD by maybe twice or even 3 times in veterans. We don't exactly know the biological mechanism for this increased risk to PTSD. We have some ideas. So we think that traumatic brain injury fundamentally is a disorder of networked brain communication. Different areas of the brain can't talk to each other very well. What happens is that the injury damages white matter tracts in the brain. These are these fibers that connect different parts of the brain and allow them to talk to each other. So you have a TBI, the fibers get severed or damaged in some way, the insulation of the fibers gets stripped off, and this impairs the ability of different brain regions that may be connected by, say, this big red fiber here to be able to talk to each other. They can't talk to each other very well. So we have this potential model where a traumatic brain injury reduces the integrity of your white matter tracts and reduces your brain communication. It's possible that these connectivity problems can underlie vulnerability to PTSD. In particular, you can think about if there are brain circuits that allow you to regulate your emotional response, and suddenly these brain circuits are damaged or severed, that could very well cause you to be more likely to develop PTSD if you also suffer a trauma. We think this is what's going on, but this pathway of effects really hasn't been demonstrated in any previous work. Why have we not been able to demonstrate these effects? Well, previous work hasn't collected enough data. You have very weird, fuzzy measures, especially of brain communication, and so you can't demonstrate this pathway. We wanted to see if we could get better measures of brain communication. So we scanned 37 US military veterans using FMRI and diffusion imaging, which allows us to look at white matter tracts. First we scanned them with a brief initial scan to make sure that they were comfortable in the scanner, make sure that we could have them in there for a while and that they wouldn't get too uncomfortable and that they wouldn't start having problems. Subjects who were comfortable came back for up to 4 additional 2-hour scanning sessions. So we wanted to scan them as much as we possibly could. In all, we had 26 subjects who completed at least one of these 2-hour scanning sessions and seemed to have good quality data. When we did the same analysis that we did in our healthy controls, we got effectively the same result. When you collect enough data, you get very reliable measures of brain function. So each one of these lines, again, is an individual participant. If we were to do the standard thing and collect 5-ish minutes of data, we don't have particularly reliable measures. What do these reliable measures tell us? Well, it's very interesting. They've really pointed towards 2 pathways for how traumatic brain injury tends to influence PTSD. In the first place, we found that the more TBIs you've had, increasing number of TBIs that you've suffered, the worse, the more disrupted your white matter tracts tend to be. And this effect does predict the severity of PTSD symptoms. And we also found an entirely separate effect where the worse the TBI you've had, so if you had a very severe TBI, that affects the communication, your brain network communication, the ability of different brain areas to talk to each other more than if you had only a mild TBI or no TBI at all. And that also affects the severity of your PTSD symptoms. Interestingly, these 2 pathways, we really thought this would be a single pathway where the white matter tracts influence the brain communication. It doesn't seem to be the case, and we're still trying to figure out exactly why. So we're really moving forward in understanding the neurobiology behind TBI effects. I think really critically to note is, again, we were able to show these pathways, these 2 pathways, with this high data scans. if we did the thing that neuroimaging has tended to do, get only a little bit of data, we couldn't see any of these. All of these red pathways became non-significant. We could not see those relationships. So in summary, we think that this high data neuroimaging approach really has helped us to understand better biological pathways by which a traumatic brain injury can affect PTSD. And this is really the first work showing that measures of brain communication link these two measures, these two conditions. We've also shown that high data scanning, getting very reliable individual specific measures, increases the reliability of these measures in clinical populations, and it is what lets us identify these links between TBI, brain connectivity, and post-traumatic stress disorder. This is still not clinical relevance. We still have not taken neuroimaging and made it clinically relevant. As informative as this doesn't provide any diagnostic or prognostic utility yet. But by figuring out how we can collect this precision neuroimaging data, we're getting a lot closer, I think. The big questions that we really need to ask next, now that we have these extremely precise measures of brain function and organization, can we use these to diagnose TBI patients, individual TBI patients, not these groups? Can we determine optimal courses of treatment using these measures of brain function? Can we find patient-specific brain areas, specific brain targets that we can target with interventions, like there are fancy new techniques like transcranial magnetic stimulation, where you can have a magnetic pulse at a specific area of the brain to affect the function of that area of the brain. Where do we need to target in the brain? We don't really know until we get these individual specific measures of brain organization. Stay tuned on all of this. We're working hard to figure out all of these questions. So I want to acknowledge in particular my collaborators at the Center of Excellence in Waco, and our collaborators at Washington University School of Medicine in St. Louis, with whom we did this work on health controls. Thank you very much.